Hi, my name is Michael Fatemi, and my teammates are Eric Feng and Cyril Sharma, and we developed RNA imputation techniques to augment spatial transcriptomics data.
So today we'll be going over what spatial transcriptomics is, methods for predicting RNA expression with deep learning, validation techniques for scarce data sources, and analyzing spatial autocorrelation. So a bit of background. Metastasis is the leading cause of cancer deaths. So predicting whether or not a tumor will metastasize is a crucial problem of research. However, just looking at a whole slide image is not very informative, so that's where spatial transcriptomics comes in. Spatial transcriptomics essentially gives you gene data at each pixel across an entire image. And this can allow you to make much more informed predictions as to whether or not it will metastasize. The only downsides is that it costs upwards of $3,000 per slide image. So the problem is spatial omics data is scarce and it's expensive. However, whole slide images are not scarce. So if you were able to go from whole slide images, if you were able to predict the spatial transcriptomics data from whole slide images, then all of a sudden you would have a lot more data to work with. And that's what we tried to do with our model. So our model just takes in whole slide images and it predicts the spatial transcriptomics associated with that data. So some related works that did similar things is he and al, which developed STNet, density model that predicted gene expression, Levy-Jurgensen, which used an inception model to predict gene expression, and Zheng and al, which used vision transformers and graph convolution networks to provide context. So now onto our model. So the first step for our model is pre-processing the image data. And so for how it works is we take an array of gene expression labels, and for each location in that array, we crop out an image of the surrounding tissue in the whole slide image, and then that patch that cropped that portion is normalized and rotated for training purposes. Our training targets, we have two training targets. We have a dichotomized training target, which is essentially predicting will the gene expression be high or will it be low. And high is defined as greater than median, low is defined as less than median. And then there's the continuous prediction, which just predicts the log normalized transform of the gene expression.
So we can treat the problem of RNA imputation as an image classification or regression problem. So for image classification, we only take into account local context. And the way this is done is by taking a cropped image centered on the location we want to predict the gene expression for, and then passing it through an inception v3 model. So we chose an inception v3 model because it has global average pooling, which reduces overfitting, as well as auxiliary logics, which enable it to fine-tune more efficiently on scarce data. These latent space embeddings will come back in the future. These output logics are general purpose and can be fine-tuned to either binary or regression models. So some of the loss functions and metrics we have. So we have about dichotomized classification, which uses average precision as the main metric because in a heavily right-skewed data set, such as gene expression data, accuracy is not informative. For the loss function for this binary classification, we use weighted binary cross entropy, which means that we take, instead of taking the mean cross entropy loss across all instances, we take the mean of the positive cases, or of the negative log likelihood for positive cases separately from the mean of the negative log likelihood of the negative cases, which means that we artificially inflate the loss for positive cases, which means the model learns something more general. For continuous regression, we use root mean squared error of log predictions as the metric, which means that we'll know relatively how incorrect the model was rather than absolutely, which is more relevant here. And for the loss function, we can model the gene expression distribution as a negative binomial distribution, and we can use zero inflation to make a model that can take into account like genes that are only present in specific parts of the Visium data. So we also train on specific subgroups of the transcriptome. So one subgroup is the 10 genes from the STNet paper, which demonstrated high contrast with tumors. And two other subgroups were from the Mouse Informatics database from the Jackson Institute, and they have negative and positive, they are associated with negative and positive regulation of T cell receptor signaling. And we chose those subgroups because we believe that they would be relevant to the tumor immune microenvironment, as well as the interaction between immune cells and the tumor. The way we use these subgroups is by taking the maximum gene expression of any gene in the subgroup and taking that to be the expression of the subgroup in total. So here are some of the results. This is for the binary classifier. On the left, we have the tumor-specific gene groups. Here, we find that the median area under the ROC curve increases with patch size, and as well as the median average precision, which is what we're interested in. And so that indicates that increasing the amount of spatial context makes it so that the model can generalize more effectively. For the whole transcriptome, we found less noticeable changes in the ROC and the average precision, but they still generally trend upwards when we increase the patch size. Here are some results from the regression model, where we're predicting log transform gene expression. So as we increase the patch size for the tumor-specific gene groups, the median R-squared value increases and the median root mean squared error also decreases. For the whole transcriptome, once again, the changes are less noticeable, and that's likely because there are so many genes. The R-squared value does modestly increase, as well as the root mean squared error modestly decreasing as we increase the patch size. All of this data was from the validation slides when we used the leave one out cross validation. Here's a visualization. On the left, we have a log scale heat map of the gene expression of the maximum gene expression for any gene associated with T cell receptor upregulation. And on the right, we have the output probabilities for the dichotomized classifier for that gene subgroup. So this shows the model demonstrates well. This demonstrates the model generalizes well to unseen data because this is a validation slide. So we also tried to incorporate spatial context in other ways. One of the ways we tried to incorporate it was through a transformer model, in which we took the latent space embeddings from the penultimate layer of any inception v3 model that we trained, and then we added a sinusoidal positional encoding to that. And then we passed that through a transformer encoder, which would allow it to selectively incorporate context from the neighboring patches, and then pass it through a prediction head, which would allow it to be used for either binary classification or regression. And for the graph convolutional network, we did a similar thing where we embedded all the gene spots with the inception model, and then we passed it through n graph convolutional layers, which each use mean aggregation, and then passed the aggregated embeddings through a linear layer and then a ReLU before being used as the next value for the node. And then we'd pass the encoded or slash contextualized node embeddings through another prediction head. So here are some of the results for the transformer. So the median AURST and the median average precision are much lower than what they were for the whole transcriptome over here. So it was 0.2 roughly for the whole transcriptome when we were using just an inception v3 model. However, when we use a transformer, it's down to 0.1284. And so a similar thing happens for graph convolutional networks, but to a lesser extent. We find that increasing the number of layers in the graph convolutional network decreases the accuracy as well as the average precision. So there are some reasons we think this might be happening. One of them is that the gene expression data is not very smooth, which means that the smoothing caused by graph convolutional layers or self-attention in general may weaken predictive ability of the model. In addition, graphs may be less useful for coarse data, which just means that the data might be less smooth than we thought it was. And another thing is that a lot of information is unseen the further away we try to integrate the context. So if you're trying to integrate distant global context, there's a lot of information, such as, for example, the tissue in the whole slide image is just two-dimensional. However, the tissue is in reality is in 3 dimensions. So the further away we get from the gene spot, the more likely there's some information, some tissue that is closer to the spot in a layer that we cannot see in the whole slide image that we just don't know about. So trying to train a vision transformer or a graph convolutional network would simply increase the chances of overfitting.
The next step in our workflow was a spatial autocorrelation analysis, but before we could pass spatial transcriptomics data through our spatial models, we needed to process the data a little bit first. So for each gene in the Visium data, we summed the expression counts to create a total counts matrix. We use this as a reference for individual gene expression to compare back to. We removed genes from our analysis that had a sum expression across all barcodes of less than 3 sequences, since those genes were so lowly expressed that they were insignificant to our analysis. And since gene expression generally follows a negative binomial distribution, we had to transform the data. So we transformed the data using an Anscombe transform, and this allows us to normally distribute the noise. And in order to further analyze the slide images and to save on computation resources, we had to decrease the resolution. We did this by simply passing a 2x2 max pooling filter over our Visium data. So the two main libraries we used in analyzing spatial autocorrelation were Spark and Spatial DE. So both of these were used to validate that the predictive models that we had made were retaining spatial information in the RNA imputations. So Spark and the computationally more efficient Spark X are our libraries that we use to identify spatially dependent genes. So these genes were judged based on combined and adjusted p-values for their significance in spatial variation. Since Spatial DE is optimized for smaller subsets of genes and smaller sample sizes, we passed only the top 1,000 genes that we got from the Spark and Spark X analysis to Spatial DE, which extracted several more spatial parameters, including fraction of spatial variance, the likelihood ratio, the p-value, and the q-value. We further analyzed Visium data using automatic expression histology, or AEH, which allowed us to group the 1,000 significant genes into 5 subgroups based on similar spatial expression. So Spatial DE's computation time, as you can see here, increases cubically for each additional imputation. So this is the reason we had to decrease the resolution of the Visium data before obtaining AEH results, which we simply did by passing that 2x2 filter. So the workflow through Spark and Spatial DE allows us to identify groups of genes that are both dependent on spatial coordinates, so significantly dependent on spatial coordinates with very low p-values, as well as predicted accurately by our models with high AUROC values. So the graph on the right shows a strong negative correlation, which suggests that significant spatial variability is predicted more accurately by our imputation model, and that our imputation model is accurately capturing spatial information. And we also plotted our AEH information onto here, and we can identify 2 histology groups, 1 and 3, that could provide valuable embeddings with predicting metastasis, since they are accurately predicted by our models and capture spatial information. So we further looked into those 2 histology groups using web interface biological pathway profilers, specifically gProfiler and Gorilla. And since both of these had high AUCs and predicted, since both of these had high AUCs predicted by the model and significant spatial dependence, they may be valuable in our further analysis for predicting metastasis.
Here we compare the imputed RNA distribution to the ground truth. So we run the spatial differential expression workflow on imputed gene expression, and then we compare the results from the imputed RNA with the actual ground truth RNA. And we found that there's a Spearman correlation, which is a metric that tells how well the 2 distributions can be modeled with a monotonically increasing function. So the Spearman correlation is 0.4999, which and it can range from -1 to 1. So this is a modest correlation here. This is to be expected. The model can learn to predict gene expression in a way that is roughly consistent with how it would be expected to be found in the wild. So here are some next steps. So next steps are we want to impute RNA as a form of synthetic data and use it for colon cancer metastasis prediction in the future. Another thing we might want to try is super resolution with cellular deconvolution, in which we assign RNA to specific cells by using known single cell RNA information. Another thing we can try is looking at individual cells and incorporating that data to predict the gene expression for a location, rather than using the whole patch image through an inception model. And finally, we can use this spatial omics data to correlate which biological pathways are most associated with metastasis. Yeah, thank you for watching the presentation.